Well, welcome everybody to the Oxford Martin School. Welcome to everybody who's here in person and to all our guests and audience online. You've come on a wonderful afternoon for photosynthesis, as the sun has just emerged. And it's a great pleasure today to welcome our guest, Professor Steve Long from the University of Illinois in the United States. Many of you will know Steve, but he's one of the most distinguished researchers working in the field of plant photosynthesis and plant productivity. I was fortunate to garner some biographical notes from a feature article to which he was subject in The New Yorker about 18 months ago. And from that I learned that he grew up in London, did his first degree in agricultural botany at Reading University, and after a brief spell at Tate and Lyle, which is, I guess, where you had one of your first encounters with sugarcane and C4 plants, you did a PhD with Harold Woolhouse. And after a distinguished stint at the University of Essex as a lecturer, Steve moved about 25 years ago to take up a position at the University of Illinois in the United States. And his career has, as many of you will know, has been thriving over the last 25 years. He's garnered many awards. He was elected to a Fellowship of the Royal Society in 2013, US National Academy of Sciences in 2019, has had a whole host of other awards. He's one of the most highly cited scientists working in plant biology today. And his work has really hit the headlines in recent years for the success he's had in attracting funding from the Bill and Melinda Gates Foundation, also from the Department of Energy and other funding agencies in the United States. And he'll be talking to us today about his research on ways of maximizing the productivity of plants through manipulation of photosynthesis. I know Steve well, but I don't think I know of any other individual working in plant biology who's quite so thoroughly dissected photosynthesis from the incoming quanta of radiation through the detailed mechanism of the biochemistry of carbon fixation through to the productivity of plants that are actually growing in the field. And I'm sure Steve will give us a wonderful, wide-ranging lecture today, and look forward to welcoming you and hearing your talk on maximizing photosynthetic productivity. Steve.
Thank you. Thank you, Andrew. It's going to be quite hard to follow that. so this is where my partner says I live. The Institute of Genomic Biology at the University of Illinois, which has all the tools you might expect of the Institute of Genomic Biology. But right in front are the Morrow plots. And Professor Morrow in the 19th century went to visit Rothamsted and saw the broad bulk and decided, well, the US needs something like that to understand sustainability of its cropping systems. And so this is the oldest agricultural experiment in the United States. In fact, I believe the oldest one outside of Rothamsted that's been in continuous operation, the Morrow plots. So although I'm in an Institute of Genomic Biology, we are quite grounded in agriculture. Today, more than 1 in 10 people on the planet are starving. So short of food for significant periods of the year, often perhaps goes unnoticed because most of that is happening in rural areas of sub-Saharan Africa and poorer areas of Southeast Asia. The World Food Programme says that number has increased by almost 50 million in just 2 years. And the forecasts are that this number is going to become progressively worse over the next couple of decades. Now, of course, some of that is caused by conflict, but a lot of it is actually occurring in areas where there isn't conflict. And of course, we've been improving the yields globally of our major crops almost continuously since the 60s. And if we can continue doing that, we might be able to carry on along this line. By 2050, we'll be here. But if you look at trends in demand, population growth, changes in diet, then we need to be more like here. So there is an ever-growing gap, which is, if that continues, will cause continued price rises in basic commodities. Now, for many developed countries, a doubling of food prices is not quite a catastrophe, but if 80% of household income goes on food, as it does in many parts of the world, it is a catastrophe. So we need to be able to find ways to up the rate at which we've been improving our crops. Now, a number of people have argued, well, actually there are other ways we can do this. We can reduce demand by diets becoming more vegetarian. We can avoid all the losses that occur in the supply system from harvest through to food that's purchased in supermarkets and then maybe wasted in the house. And then, of course, we have the elephant in the room on top of this, climate change. Well, if we look, however, at, for example, meat consumption, that has been rising steadily since the 60s. In fact, it's almost doubled per capita globally in the last 60 years. And there's no sign of us getting to peak meat. If we look at wastage, then the World Bank has forecast, based on current trends, and particular trends into urbanization, where more and more food has got to be transported from the countryside to cities. Their forecast is that waste is actually going to go up, not decline globally. Now we have another issue that, although we've really had quite spectacular increases in crop productivity, maize in particular, in the US Midwest, has almost tripled over 100 years in yields. But what we haven't done with that maize is to improve water use efficiency. So if we double or triple production, we're also doubling or tripling water use. And what we forecast here is that for this to continue, by about 2030 or 2040, for example, in the center of the Midwest, about 10% of maize is irrigated. To get continued increase, we would now have to move to 50% irrigation. And agriculture is already accounting for about 3/4 of human water use. So we need to be able to increase productivity without increasing further demand, particularly in terms of water. Now, if we look at what determines the yield potential of a genotype on a given piece of land, if it has the water and nutrients it needs, we defend it from pests and diseases, then it's determined by how much solar energy is available over the growing season, how good is the crop at capturing that solar radiation, how good is it at converting that captured radiation into biomass, and then how good is it at partitioning that biomass into the parts of the crop we care about. So the grain of wheat, the seed of soybean, and so on. Now, if we look at our major crops before the Green Revolution, interception efficiency in those crops was around about 60%. Partitioning efficiency or harvest index was around about 30%. If we look at post-Green Revolution, then we see harvest index has been improved almost up to what we think is the theoretical maximum. So modern wheat cultivars are partitioning 60 to 65% of their biomass into the grain. So if there's going to be some stem and structure to hold that grain, it doesn't seem likely we can get much further. The same with rice as well. Corn is a little bit, maize is a little bit behind that. We've also managed to improve interception efficiency. So modern cultivars are faster getting out of the ground, better at covering the ground, faster so they capture more of the solar radiation available. But if we come to conversion efficiency, current cultivars and pre-grain revolution cultivars show very little difference. A tiny improvement in wheat, in soybean. And conversion efficiency, of course, is primarily photosynthesis. How good is it at using that radiation and converting it into biomass? Well, what we're seeing since the Green Revolution is something of a slowdown in yields of some of our major crops, yield improvement. If China, India, and Indonesia are the biggest producers of rice in the world between them. If we look at the Green Revolution years, post-Green Revolution years, 70s and 80s, they were averaging improvements of more than 30% per decade. If we come to the most recent 2 decades, then what we see in China is this is down to single figures. And what's remarkable here is that China now is spending more than 100 times, in real terms, on rice improvement that it was spending in the 70s and 80s. So it's telling us it's getting harder and harder to get these improvements. Now, the UK is in the top 4 for its yields of wheat per hectare of land. And between 1970 and the late 1990s, there was quite a remarkable improvement in wheat yields in the UK. Average yields improved from 4 tonnes per hectare to 8 tonnes per hectare. But then that seems to have stopped. And despite all the improvements in our knowledge of the genetics of wheat, identifying many different genes for improvement, and of course there have been big improvements in tolerating new diseases arriving, we're not seeing an increase in yield. We are seeing more yield instability, part of which may be related to climate, part might be related to genetics. Similarly, France seems to have kind of hit a ceiling. France has put a lot of effort into mixtures of different cultivars to try and get a bit more yield stability, although there may be some evidence of that occurring, although INRA, who've been behind this, say they don't have the proof of this as yet. So coming back to photosynthesis, could this be one means, and I'm not saying it's the only means, but one means that we might be able to start lifting what appear to be these ceilings? Well, first of all, we need to know kind of what is the theoretical efficiency of the process. Photosynthesis is probably the best known of all plant processes. We know every step in the process. We know a great deal of detail of the proteins involved, the genes coding for those proteins, the kinetic properties of those proteins. So we can really go through the thermodynamics and say, well, for 1,000 kilojoules coming in, using the existing process, what should come out if it's perfect? Well, that tells us that in the current atmosphere, with a C3 crop like wheat, we should be getting out about 4.6% efficiency for total radiation, with about 10% for visible radiation. Our best crops, however, are achieving probably about 1%. So that tells us there might be a lot of headroom here if we could improve this. C4 comes out somewhat more efficient, 6%. If you go to visible radiation, that may be about 12.5% theoretical. So these, of course, are 2 heroes of the Green Revolution, Norman Borlaug with wheat, Yuan Longping, one of the rice breeders of the Green Revolution. And I was lucky about 10 years ago to be able to meet him, and behind us here is one of his rice cultivars. You can see here's the flag leaf. The panicle is coming right down to the paddy surface. What he's complaining to me about is, in his latest cultivars, the caryopses, the seed, are not filling. And he's saying to me, when are you guys going to get your act together and give me more photosynthesis to start filling these seeds? Well, okay, we haven't improved this through breeding, so what makes us think there's any chance of doing it now? Well, as I said, photosynthesis is the best known of all plant processes, and it's fairly uniform between all C3 crops. We have high performance computing, which allows us to simulate the process. And we now have crop genomes revealed in great detail, and crop engineering is becoming ever increasingly more efficient. But if you look at what happened, you analyze what happened in the Green Revolution, many of those changes, particularly the shortening of stems, so that more biomass could go into the grain, you could apply more nitrogen, so on, involved changing just a few genes. Photosynthesis is a process of about 160 steps, all quantitatively variable. So each facilitated by a different protein, many of them coded for by a number of genes. So where do you start? Well, one way would be to look at it like a car production line. CO2, water and light in, carbohydrate out. And so when I moved to Illinois and we had a supercomputer, that was what I thought we would try and do. Everybody said, you're crazy, which I already knew. But fortunately, a graduate student, Jingwan Zhu, who joined me with a computational background, wanted to take on the challenge. And what the challenge was to represent every one of these steps by a differential equation. So for many of the enzymes, there were Michaelis-Menten equations, but that doesn't apply to all enzymes. So we spent 3 years building all of these equations. So we got a system. The idea was we could numerically integrate that. And then once you got numerical integration, you can start changing with a high performance computing, changing each step to see whether you can get an improvement. Well, after 3 years, we were ready to do our numerical integration, and that failed, and it failed miserably. But luckily, at our computer center, another scientist said, oh, I had exactly the same problem trying to balance rocket motors in my NASA project, and this is how I solved it. And he looked at what we were doing. He was a mathematician, and he said, oh, this is much more challenging than what I'm doing. I want to work with you on this, which is why we have a rocket scientist as the second author on our first paper. So it's just to point out that photosynthesis isn't rocket science. It's quite a bit harder. But, and of course, with plants, you've now got to take account of the fact their environment is changing all of the time. So light changes over the day. As leaves develop in the canopy, they start off in full sunlight and get deeper and deeper into shade. But after a few years of doing this modeling, we started to pick out a number of ways in which we thought, from modeling, of course, how we could improve photosynthesis and make predictions on what gain we might get, 15%, 30%, from these models, and how long that might take. So at this stage, funders started to get interested. But one of the questions was, well, if you could do this, if you could improve photosynthesis, how do you know you're going to get more yield? Well, that made us go back to an experiment I was able to start when I first moved to Illinois, and others have taken it on to much greater heights. But these plots on our university farm, the university is over here, and then the farm extends out. Each of these plots is a ring like this, where we can release carbon dioxide according to wind speed and direction, so that within the plot, the CO2 level is elevated to what we think the level will be in 2050, 2060. Now, what elevating CO2 does is to boost photosynthesis because it causes an acceleration of carboxylation and an inhibition of oxygenation, which occurs in the primary carboxylating enzyme. So there's a lot of knowledge that boosting, if you could raise CO2 without altering anything else, you will get more photosynthesis, more productivity. And of course, this is why greenhouse growers elevate CO2 in their greenhouses. So when we did this in the open air, we could see without soybean, we were getting 16% more yield. In wheat, we were getting 15% more. And then experiments done in China and Japan using replications of this technology, we're also seeing yield increases. So that told us if we could boost photosynthesis, we would get more productivity. Now, with more years of this, we were able to look at different cultivars, and experiments in Japan and China did the same with rice. And interestingly, what we saw was this boost in productivity was much higher in the current elites, high-yielding cultivars. The higher the yield potential, the bigger the response to boosting photosynthesis. So we were able to kind of present this in saying, yes, if we could boost photosynthesis, we get more yield. And so, well, the funders got interested and more or less said, okay, we're willing to put our money where your mouth is. This was the Gates Foundation, first of all. And you show us this can be done. Well, of course, you can do great things with models. They look very attractive. But now you've got to show this will work in a muddy field. It's not quite so easy. And there were many skeptics saying, this is never going to work. But nevertheless, we managed to get together a team of scientists globally who were willing to go where no sensible plant biologists had been before. And this is Chris Niogi from Berkeley, Susanna von Kamerer from ANU, TJ Higgins from CSRO, Christine Rains from the University of Essex, Martin Parry, who was at Rothamsted when we started this work. So we moved forward on a few ideas, and this is the way we've been working. So we use the modeling to predict what we think will be the simplest changes that could give us a big increase. And then we engineer that into tobacco. And you might think, tobacco? Why on earth is the Gates Foundation supporting that? The reason we use tobacco is that it's very easy to genetically transform. And it produces a lot of seed. So basically you can go in 1 year from transformation to a replicated field trial. In soybean or wheat, would be about a 4-year process. But tobacco allowed us to filter out, does it really produce more productivity in the field? If it does, it's worth going to the crops you care about. And the Gates Foundation was particularly interested in us doing this in soybean and cowpea. Cowpea might not be what you think it is. It's a leguminous crop as well. Very important in sub-Saharan Africa. And our final test is, okay, in such a crop, does it result in improved productivity on our farms? So I'll tell you about 3 successes we've had, and then I want to briefly mention C4 photosynthesis. So the first of these was upregulating chloroplast C-deheptulose bisphosphatase. This is an enzyme that's involved in the regeneration of the molecule which accepts carbon dioxide at the primary carboxylase, rubisco. Rubisco, incidentally, is the most abundant protein on the planet. So every plant uses this for assimilation of carbon dioxide. And this enzyme is involved in recycling its CO2 acceptor molecule. Now, our digital twin predicted, well, this enzyme should be far more abundant than it is. So this is what the plant has. This is what the digital twin said it should have to optimize photosynthesis. So Christine Rains at the University of Essex, been working on SBPAs for a while, antisense originally, and then, so she put additional constructs into tobacco, and in the greenhouse, these were much bigger plants. Now this was a bit of a puzzle because this enzyme is a tiny fraction of leaf protein. So why hadn't evolution already done this? Well, a former student was visiting us when we got these results, Colin Osborne from the University of Sheffield. And he said, well, probably the crop ancestors evolved in a much lower CO2 concentration than we have today. Over the previous 4 ice ages, the average CO2 concentration of the atmosphere was about 220 parts per million. Today it's 420 parts per million. So we reran the model at 220 parts per million, and under those conditions, this enzyme didn't seem to exert control. But as you increase CO2, it exerted more and more control. The other thing we realized was temperature also tended to increase control. Now, we couldn't take away CO2 in the field very easily, but we did know how to add it. So we took Christine's plants and put them in this CO2 facility, and sure enough, as the model predicted, we got an even bigger improvement when we upregulated this enzyme. Now, another former student, Carl Vanacki, developed an open-air heating system where basically, so he's imaging the surface of the canopy thermally, and then he adds in 5 degrees Celsius to keep a constant temperature elevation. And when we did that, again, we saw a bigger difference between our engineered plants and the controls. However, in this case, it was protecting the plant against future conditions. It wasn't resulting in more yield. It was just managing to maintain that yield. So one that my lab has been closely involved in is accelerating this, a relaxation of non-photochemical quenching. And I'll explain what that is in a moment. During the day in a crop canopy, the top leaf might see constant illumination as the sun crosses the sky. But our modern crop canopies are 4, 5, 6 layers of leaves. And as the sun moves across the sky, this means that the leaves below that are going in and out of shade all day long. When that happens, photosynthesis drops, of course, when a leaf goes into the shade, but it drops much lower than it needs to. And then it slowly recovers, and it's doing this over and over again. So all of this is lost assimilation. And if you add that up over the course of a day, it can be 20 to 40% of potential carbon fixation. So essentially, what is happening is when a leaf is in full sunlight, it's absorbing more light than it can use. If that excitation energy is not removed, oxidizing radicals are produced, which would literally destroy the leaf. So evolution has dealt with that by evolving this process of non-photochemical quenching, where it diverts that excess energy into heat. And so as soon as the leaf goes into full sunlight, this non-photochemical quenching comes on stream, and then when it goes back into the shade, it relaxes. But it relaxes rather slowly. It's rather like you being in the sun outside, you walk into a room, and it takes a little while for your eyes to adjust. And it's the same with photosynthesis. Instead of adjusting in maybe 30 seconds, it can take 15 minutes or longer. So it carries on dissipating heat, even though now it could use all of the light it's getting. So it takes many minutes before that process relaxes. So from our modeling, and also Chris Niogi's work on Arabidopsis, we were able to deduce 3 proteins we should be upregulating. And Laurie Leonelli, who's now an assistant professor at Illinois, but was Chris's postdoc, developed a construct, and Kasia and Vanna Kromdijk, who's now at Cambridge, put this into tobacco, and we were able to get tobacco plants which were expressing all 3 of these. And we could see that the non-photochemical quenching was relaxing faster. In the shade, they had more photosynthesis. And in the greenhouse, these plants look bigger. And eventually we were able to demonstrate that in the field, and we were getting between 14 and 21% more productivity in tobacco. And Science put that on the cover, except strangely, they put a soybean plant on the cover. We figured this because they don't often deal with crops. But it was prescient because that was where we wanted to go next. So I should add, we also looked at the individual genes, and one of the things we noted was that upregulation of PSPS also gave us a significant improvement in water use efficiency. So the plants were using about 15% less water at the leaf level per molecule of CO2 taken up. So eventually we were able to do the same thing in soybean, and we could see again in these plants that they were able to relax their non-photochemical quenching faster, and we were able to see a significant yield increase, i.e. seed yield increase in soybean. And what was particularly pleasing with this was that if we looked at the protein content in these, we saw very little change. So that meant the protein content of the seed was unchanged, even though there was 20% more seed. So that meant the plant was able to get its nitrogen from somewhere, because in line with agricultural practice, we don't add nitrogen fertilizer to soybean, it fixes its soybean. The only way it could have done this was to have partitioned some of the extra photosynthate to the bradyrhizobium to fix the nitrogen to be able to maintain the protein content in the seed. So this suggested for legumes, this could be a sustainable increase. In 2021, however, we repeated the experiment with bigger plots. The crop was lodged and we didn't see any improvement. Amanda, who conducted this work, also put our best line into soy phase because we were curious. Well, if we're already boosting photosynthesis in soy phase by elevated CO2, would a genetic transformation that boosts photosynthesis still result in an improvement? And indeed, what she saw was that there was a boost in both ambient and elevated CO2, and there wasn't a significant interaction. So it suggested, even if you were adding on other ways of boosting photosynthesis, you could still get a yield increase. Now, again, you wonder why might this happen? And one reason might be that, if we look at the wild ancestors of wheat or soybean wild ancestor is a climbing plant, usually growing in fairly poor environments where there's going to be very little shading of leaves. Whereas today we're growing it in these dense crop canopies where shading will be very frequent. Similarly for wheat. So it could be that we've taken some plants and now we want them to behave well as sun and shade plants. And indeed, Chris Niogi has now been looking at a fern growing in a forest understory and shown that it relaxes its non-photochemical quenching very rapidly. So, and of course, we're now looking at how it does that, whether we can transfer that to crops. So I want to talk about a third way in which we managed to get a big increase in yield, well, in productivity of tobacco. Is now being trialled in food crops. Photorespiration reduces the productivity of C3 crops considerably. But that is due to the fact that rubisco, this highly abundant protein on the planet, not only does it carboxylate this primary acceptor, ribulose 1,5-bisphosphate, it can also oxygenate it, which leads to the process of photorespiration. So some of this carbon is recycled, some is lost as carbon dioxide. And not only is this sort of occurring, but it increases with temperature. So with climate change, we think losses due to photorespiration will get larger and larger. And at 40 degrees, more carbon is lost to photorespiration than the net gain. So if you can lower the cost of photorespiration in any way, it will really help in future-proofing crops. Now, this recycling of the product of oxygenation of rubisco is, in higher plants, goes through the peroxisome, the mitochondrion, and eventually back into the chloroplast. In doing that, it's using a lot of ATP and NADPH. And so what this team, headed by my colleague Don Ort, have been doing is looking at alternative recycling pathways that will use less energy. And this work was conducted by Paul South, who's now an assistant professor at Louisiana State, Amanda Kavanagh, who's a lecturer at University of Essex now, and Catherine Meacham, who is now the science manager for the RIPE project. And what they did was to put in target enzymes to the plastid to recycle the glycolate, this product of oxygenation, and to also close down, we're using RNAi to reduce the amount of that glycolate that was going out of the chloroplast. And cutting a long story short, they showed that in the field, they could get about a 30% gain in biomass by reducing these energy costs. Now, Amanda Kavanagh noted in her photosynthesis measurements, as we predicted in the modeling, that the benefit of this increased with temperature. So here is the wild type, here are the transformants. So at 40 degrees, a very large difference. Now, of course, we could go back to that field experiment where we can elevate temperature over the growing season. And so this was elevating the temperature of a soybean crop throughout its life by 5 degrees, or sorry, not soybean, tobacco. And what you can see here is that 5-degree elevation of temperature hugely lowers the biomass of tobacco. We know it does the same in soybean. And these manipulations resulted in smaller losses. Now, how does all that connect to what I was saying at the beginning? That the people in the world are starving. And surprisingly, a large number of those who are suffering starvation are smallholder farmers in sub-Saharan Africa, in Southeast Asia, feeding their families from 2, 1, half a hectare of land, often half a hectare of land. We do know that one of the best ways to lift people like that out of starvation is to get them seed with more potential. Whether it's defending it against pests, diseases, or simply giving them material which has higher productivity. So what I've shown you will be great for the Midwest, etc., getting higher soybean yields, etc., but can we get it into crops which really count, where it's most needed? Well, we've been working with TJ Higgins from CSRO in Australia, who has pioneered with breeders in Nigeria, Ghana and Burkina Faso, trying to get insect-resistant cowpea in place in those countries. And so he's been working with them to put the Bt genes into cowpea. Bt is a bacterial gene which is produces a protein which is very toxic to a large number of insect groups. So it kills off many boring insects. For example, if I say boring insects, I mean not ones that aren't very interesting, but ones which bore into the stems of crops, bore into the pods of cowpea in this case. Cowpea is very important in West Africa. It's often called the poor man's meat because it is the source of protein for many families. And a pod-boring insect can devastate a smallholder's crop, can completely wipe it out. Well, in 2019, we're developing regulatory processes with Nigeria. It was eventually possible to get this Bt cowpea into the seed system in Nigeria, where it's become very popular, very effective. And last year, it was deregulated in Ghana as well. So what we're trying to do now with TJ is working with us, is now putting our traits on top of his Bt resistance. And all of this will be available royalty-free to farmers in these countries. And so we're now trialing this in cowpea in Puerto Rico, where we have, Illinois has an experimental farm, but it's a tropical climate. As it's part of the USA, it makes it that much easier to conduct these trials. So finally, one of the questions you might say, well, what about C4 photosynthesis? That's already very efficient. Could you possibly improve that? And of course, it's very important for food crops, maize and sorghum, but also for bioenergy crops, energy sorghum, which is incredibly productive, sugarcane, energy cane, which is derived from sugarcane, and one of my favorite crops, miscanthus, as well, which is highly productive on marginal land requiring very few inputs. So can we improve on these? Well, Yu Wang, who was a postdoc with me, built a model from our C3 model to NEDPME C4, and then we numerically integrated that and start to probe where are the pressure points. And two pressure points that came up were pyruvate kinase, and again, this enzyme Rubisco. The model was saying there isn't enough of it. We also looked at it under dynamic conditions, and that came up with two more proteins, the activators of these two enzymes. So another postdoc, Coralie Salisi-Smith, working with me, built a construct to upregulate Rubisco in sugarcane and in sorghum. And on the left here is Freddie Outpeter, professor at the University of Florida in Gainesville, who has been transforming sugarcane. So he took our construct, put it into sugarcane, and we got lines that were expressing both of these proteins. And Coralie's examined these, and you can see here that these are ACI responses. She's getting photosynthetic rates, which are already very high in the wild type. But in the transformed plants, she's getting a 10 to 6% increase on top of that. And it's also where it should be, because Rubisco limits maize photosynthesis at high internal CO2 levels, but not at low CO2 levels. So it fits what we've been modeling. And so it does show that we can increase photosynthesis. We're waiting to see if that improves productivity. The other thing that Yuang predicted was that in fluctuating light, activation of rubisco in C4 plants would be a major limitation. So it'd be the slowest component to activate on a shade-sun transition. And she came up with a way we could test that, because what happens in C4 photosynthesis is the outer layer of photosynthetic cells act as an energy-driven pump. They take CO2 from the atmosphere and put it in this inner layer of cells where rubisco sits. And what this does is to concentrate CO2 at rubisco. CO2 is a competitive inhibitor of this oxygenation reaction. So by elevating CO2, C4 plants avoid photorespiration. That gives them their higher productivity than C3 plants. But if this is slow to activate, CO2 will build up here and it will leak back into the mesophyll where it has to be pumped again. Well, in the natural atmosphere, we have CO2 in two forms, 12 CO2 and 13 CO2. And if you keep recycling that, there's discrimination against 13 CO2. Diffuses a bit more slowly, it's slower in reactions, and so you're altering the isotope balance. And you can pick that up in the air exiting a chamber. And indeed, we can use that to measure the degree of leakage which is going on. And sure enough, what we saw was this spike in leakiness, which gradually tailed off as photosynthesis increased. So that proved to us that upregulating not just rubisco, but its activator would be important. So I should say this instrument we use, which is a tunable laser diode, it's about 6 foot long. It came in a box, slightly longer than 6 foot, which sat outside the lab for a few weeks. And somebody put a post-it on it saying, Steve's exit plan. But anyway, so what this showed was we need to upregulate rubisco activase in bundle sheath plastids. So I'd finish by saying photosynthetic efficiency can be improved to increase productivity in food, feed, and energy crops. What we've shown in crops applies, we believe, to trees, and a couple of companies are now trying this. Photosynthesis had largely been ignored as a means to improve productivity, but I would stress it's not a replacement for everything else. It needs to be an addition to what we're already doing. All of the examples I've given you are transgenic, but many of these actually are upregulation of proteins, which could be improving, could be done by editing promoter regions. It does require an adequate sink, our unique cultivars which are going to be able to use that additional photosynthate. It could be used for other purposes. More carbon soil sequestration, for example. You could divert more carbon to roots, products like suberin, which break down more slowly while maintaining crop productivity, or it could be used to power nitrogen fixation. Another Gates project is to try and engineer nitrogen fixation into cereals, headed by Giles Oldroyd at the University of Cambridge. Well, this could provide the power for something like that. Could improve the productivity and economic viability of energy crops for marginal lands. And I've just given you a few of the opportunities we think there are in improving photosynthesis, which could, of course, be stacked. So with that, I've finished, and I think it's probably been obvious that this is the result of the work of not me, but a huge team. who've worked with me over the years, conducted much of this work. So thank you for your attention.
Well, thank you, Steve, for that extremely extensive and wide-ranging talk. I'm sure it'll have stimulated a lot of questions. I hope people are still able to hear us online. And we'd like to throw the talk open to a Q&A session if you're prepared to field some questions. And just for the benefit of those who are asking questions, firstly, it would perhaps be helpful if you could just briefly mention your name when you're asking a question, but also please be aware that the fact that the Q&A session is also being recorded and transmitted online, just so we're able to conduct the questions appropriately. And we'll try and take some questions, I think, from the online audience as well as they arise. Is there anybody who'd like to kick us off after that very impressive presentation? David.
David Walton from Cambridge. Picking up on the final point you made about bioenergy crops, biofuels crops. So when we had, let's say, biofuels hysteria a decade or so ago, rather famously, some of the analyses indicated that the energy input of growing those crops was actually greater than the energy output due to the costs of fertilizer production and so forth. Do you think that introducing your innovations would be able to mitigate that problem and give us actually truly productive biofuels crops?
I guess a short answer would be yes. I would say, you know, at least the analyses today are showing, like Brazilian sugarcane ethanol is really very CO2 efficient. You know, I think it's about 1/15 the emissions if you do a whole chain analysis of using petrol. But corn ethanol is a lot worse. It's probably only about, you know, Argonne National Lab's latest analysis suggests it's about 40% better. But if you come to crops like miscanthus, then, which are perennial, if they're harvested, for example, you know, the way to harvest it is you wait for it to dry down. It's translocated as nutrients into the perinating rhizome system. Then the fertilizer cost of something like that is very low. We have a stand which was the first one established in the United States, at least for agricultural trials. It's been there 20 years, and it's still highly productive. And we fertilized it with 50 kilos per hectare 20 years ago, and we've never added anything since. So I do think, you know, that these crops really could be efficient, although I think the biggest value would be using them with combined combustion, capture and storage, because that could be a real way to remove CO2 from the atmosphere, providing, of course, you've got the geology to capture the CO2. And if you, suddenly we've got a Department of Energy group who've worked through all the costs of doing that, and they claim that will be negative in terms of CO2. So part of the energy you generate, of course, is used to liquefy and pump the CO2, but you're actually net removing CO2 from the atmosphere with crops like that. So I hope that answers it.
Yeah, thanks very much.
Yes, Angela, please.
So Angela Carp from Rotherham. So Steve, you talked about some research on plants in shady areas, but are you doing the inverse? Because in the future, we are going to struggle with too high temperatures and potentially too much sunlight in parts of the world where we're not used to that. And I just wondered if any of the research you're doing is looking at how plants cope with that and what happens to their photosynthesis at times when it's too hot and there's almost too much sun. And a sort of associated question is, I know we've changed the architecture of many of our crops to increase their light capture.
Yes.
Are you also looking at that in relation to the future? and how we might either enhance or even change architecture to improve photosynthesis, but protect our crops in those what could be now hotter temperatures and drier periods that we're going to have to live through?
Yes, I guess the first question, a couple of questions in there, but the one about architecture, in this project, we do have a group headed by Lisa Ainsworth that is looking at architecture of these crops. And we have done a lot of modeling work around that as well. You know, that, actually making crops lighter in color, has a surprising benefit in terms of, because if you're changing the albedo, this is making it a little bit cooler, but you're not losing much in terms of photosynthesis, because those upper leaves are often, as you said, receiving more sunlight than they can use. And so we have been looking at that. We have been engineering that. We've also been looking in germplasm collections, because there are ones with lighter leaves there as well. And so we have been thinking about how do you adapt it to these situations. The other thing is we've also been looking at what is the optimum canopy form. And in fact, one of the things the model came up with early was with our soybean crop, which we're very proud of in Illinois because we get the highest productivities in the world. It's producing 5-6 layers of leaves, and we're thinking, is that optimal? And the model said it isn't. There's too many layers of leaves there. So we were busy thinking about how can we deal with that genetically. But the postdoc who did the modeling was an engineer. And while I and my colleagues were talking about, what genetic manipulations can we do, he organized a group of undergraduates to work with him. He went out into the field, 4 plots where they removed every third emerging leaf and got an 8% improvement in productivity, significantly higher. So that told us, okay, our soybean canopies are not optimal. The model really was right. So on the temperature issue, certainly we think decreasing energy loss in photorespiration that Don Ort's lab is doing is going to be very important for trying to reduce the impacts of high temperature. We do know that the SBPAs as well, that Christine Rains has been working on, that also appears, upregulating that appears to protect productivity a bit against rising temperature. We don't have a complete solution for rising temperature, I'm afraid. So, yeah.
Yes, I think there's a question just at the back there. Okay.
Thank you. I'm Yushi from New College here. So on your slide about the thermodynamics, where you show there is 66 kilojoules loss due to inefficiency, and around 513 kilojoules loss from outside of the photosynthetic range. Do you think is more? Do you think have greater return on the engineering by having plants can use light outside of the current range, rather than engineering the part where the inefficiency is lying?
Yes, I mean, we are, because what I showed in that figure was where we are now. Like the work that my colleague has done on photorespiration, if you put that in, that would up that number. But you are right that we might be able to make use of a bit more of the spectrum. We know that there are other systems which can use longer wavelength energy. There's higher plants have what we call 2 forms of chlorophyll, what we call A and B, but there are a range more. And we know that there are chlorophyll D and chlorophyll F that can go out as far as 800 nanometers. So we could capture probably about 10 to 15% more of that spectrum. We think that would be quite a big ask to engineer that into plants though, but we are, in our modeling, we are trying to work out what sort of benefit that might give us you know, and how practical it is. There's a group at Imperial which has done quite a lot of work on these, and we're working with them at the moment to try and work out, is it worthwhile? Is it feasible? But, yeah, good question. Yeah.
And I think we might have one or two questions from the online audience. Charles.
So the online audience is able to vote, and the highest voted question is, what role could gene editing play? And you talked a bit about that at the end, but perhaps could you elaborate on that a bit more, and perhaps say something about the different regulatory frameworks between traditional GM and gene editing?
Yeah, well, as I'm sure most of you be critically aware that I think it's about 3 weeks ago, that Britain actually passed a law which would allow single base editing, as I understand it. And for example, if we take the upregulation of SBPAs, this enzyme involved in the production of the acceptor for CO2, you know, that was just a single upregulation. Now, usually in the promoter regions, which determine how much of the gene is expressed and when it's expressed, there are often these elements we call repressor elements. So you could, with a single base pair change in the DNA, you could knock those out if you know where they are. And so you could get what we've done by inserting extra copies of the gene. You could get the same thing by changing the promoter region so that you get more of the native protein. And I think certainly under US regulations and regulations in Brazil, Argentina, and Canada, that would pass as non-GM, because the thinking is you could do this by mutation, which is mutation breeding, which has been going on for 60 years or more. This is just a more precise way of doing it. So does that answer the question?
And I'm going to abuse this by asking a question myself. I was fascinated how you interpreted the results of the non-photochemical quenching by going back and looking at the ecology of the ancestral. Now, this may be fanciful, but do you think that if you went back and looked at the ecology of the nearest crop relatives, you might get some other insights into constraints that crops have that might be manipulatable?
Yes, I think so. We have started to do that with some traits. One of the ones we were curious about is this thing called mesophyll conductance, which is how easy is it for carbon dioxide to get from the airspace in the leaf to the enzyme I keep talking about, rubisco. And we thought, well, you know, because if we can improve this, it will give us more photosynthesis and it would improve water use efficiency. So it's kind of almost a holy grail. But we did think, well, maybe domestication. Has domestication improved this? And so we did go back to soybean relatives, and it looks as though they had a greater resistance to this CO2 movement. But of course, it also raises, can we go back to those wild relatives? Might we be able to find what we've managed to achieve transgenically and do it through conventional breeding? Yeah.
Could we go to Dale Sanders at the front, and then we'll come to people at the back.
Dale Sanders, University of York. Your metabolic modelling studies have been incredibly useful in pulling out key bottlenecks in a lot of those processes that you've described. And I guess you've addressed the issue of improving those parameters you want to improve largely by changing expression levels of those key enzymes.
Yeah.
What do you think about the prospects of changing the properties of the enzymes themselves? And I'm thinking very naively here of rubisco and changing carbon dioxide to oxygen efficiency, but SBPAs and enzymes like that, are there possibilities?
I think, yes, I think there's a definite possibility of doing that. I mean, in two ways. The group at the University of Lancaster have been looking at kind of existing diversity, which might not have been exploited. But another thing that's now possible with ever bigger computers is to atomistically simulate that enzyme in action. So literally every atom is vibrating. When it reacts with ATP, you can see the conformational change. And then what that allows you to do is work through changing, residue by residue to see if you could hit on a residue which would just give you this higher efficiency. So, I mean, that will be a much more effective way of doing it than we're doing it now, and it could be classed as non-transgenic as well. But I mean, the computational power to do this has only really come about in the last few years. I have a colleague who's been doing this with nitrate transporter, and he worked with Google, and during Google's sort of low period, he was using all of their computational power to just simulate this one transporter. But it revealed a change in the protein that hadn't been known before and has now been shown. But with increasing computational power, I think what you're saying is a real way forward. Yeah.
I think there are a couple of questions from the back. If please go ahead.
Sure, Richard White from Oxford. 2 questions about bacteria. First, can you contrast the photosynthetic efficiency of bacteria that can perform photosynthesis, and are there lessons there from bacteria? And the second is, do the bacteria that grow with the plant, such as in rice fields, do they change the efficiency in any useful way?
When you say photosynthetic bacteria, do you mean, because we have photosynthetic bacteria, for example, in anaerobic environments, they're using H2S rather than water, or using propionate. So they're using other sources of protons, if you like. Generally their efficiency is lower, but blue-green algae, though, prokaryotes, of course, they are quite efficient because they've overcome this photorespiration process by having a carboxysome, a tiny structure which encapsulates the rubisco and carbonic anhydrase. So that serves as a CO2 concentrating mechanism, which almost eliminates this photorespiration. So they are at least as efficient as C4 plants, if not more. One of the things the group at the Australian National University were trying to do is to engineer these into the chloroplast of a higher plant, and they have managed to do that, but there's still more components to add. Yeah. Good.
I think we'll take one more question from the audience.
Thanks. So in one of the last slides, you looked at, I think it's leakage, or you measured the leakage with the C13 isotope, I think it is. And I was wondering if you could use plants that have a greater affinity towards, I think, C13, or just isotopes more generally and broadly. Would that be a possible way to increase, photosynthesis?
So you're saying, could we use isotopes to investigate?
Yeah, or basically select for plants which have a greater affinity towards different carbon isotopes.
Oh, well, I'm going to say there is discrimination against 13 CO2 versus 12 CO2, but it's in the thousands, so it's quite small. It's a useful signature for investigating who's assimilating CO2, and we have a world expert here in the audience as well on this, but we're really using it just as a diagnostic tool, but if we made it less discriminatory, it wouldn't make much difference, to be honest. But,
okay, no, cool.
Good thought.
Now, Charles Godfrey tells me he's got 2 very left field questions, which might be appropriate to round off the question session with.
Charles, I've turned you on.
Thank you, Clara. This is from Garth W, who's asked a series of questions. And the first is, do you see in the future, as we get to know photosynthesis, the mechanism even better, that it might be able to abstract it and compete with photovoltaics in electricity generation? And I'll do the other one so you can pick. And I have to say I don't understand this question, but Garth says that there's a recent paper, and he quotes a physics paper, suggesting that a fifth state of matter could be involved in photosynthesis, an exciton condensate. And he wondered whether you had any comments on that.
I'll take the first one. So, can you remind the first one again? Yeah, no, I think, you know, if you look at the efficiency that I showed, plants, when it gets to charge separation, you know, so you capture the radiation, do the charge separation, it is very efficient, comparable to photovoltaics. But then you lose a lot of energy in trying to get to the stored energy in carbohydrates. So all of the metabolic steps are costing you a lot of energy. And the two photosystems has certainly inspired people in developing artificial solar cells where they're using more than one receptor. You know, that has increased efficiency in solar cells. But I'd say there were early attempts to try and mimic what chlorophyll does, but I think now there are much better synthetic ones where you don't have to worry about an organic molecule remaining stable for many years. Exciton condensation, I'm sorry, is beyond me.
Well, we're going to wrap up in just a moment. I'd just like to express my own thanks to Steve and to say that for those of you who are able to stay with us and join us for the drinks reception afterwards, we've got a few posters displayed next door on one of the programs that's being generously funded by the Oxford Martin School on dry land bioenergies. We've been involved in some work looking at plants almost at the other end of the spectrum, not the super producers that Steve has been talking about, but the super drought tolerators, the plants that can eke out an existence in extremely arid environments and the uses to which they could be put in terms of biomass feedstock for bioenergy production or other uses. So that might complement the picture. But I'm going to hand over now to Charles just to say a few words.
Just very briefly to thank Andrew for chairing today, to welcome our guests to the Martin School. It's great to see Giles, David, Angela and Dale here. And Steve, thank you for such a fabulous talk. And it is quite extraordinary what you, your group and your colleagues have been able to achieve over the last couple of decades. Great that you were able to come and tell us about it. Do join us next door for a glass of wine.
Yeah, thank you everyone for coming. And I would add, we're losing our postdocs very rapidly to positions like at Cambridge. So if anyone's interested in joining us and wants to be crazy, you know, let me know.
Well, we'll make a deal. If you redirect them from Cambridge to us, then we'll send you some on the way then. Thanks very much, Steve.